This week, we'll be talking about the ways in which brains have evolved. We'll also be discussing brain development through the fetus as well as into adolescence. And then lastly, we'll touch on the aging brain in the second half of this week. We'll first talk about how brains have evolved and what makes our brain unique. If you were given the task of building a human brain, how would you create an organ that detects all environmental information, monitors all internal physiological systems, looks at both the past and future to determine the most adaptive responses at any moment, and then works efficiently and effectively? It seems impossible, right? Even building a simpler brain like the lizards, it seems like a very daunting task, but that's exactly what evolution did. But it's important to keep in mind that rather than building a brain from scratch, each time a new species evolved on this planet, what evolution did essentially was tweak the brain slightly to the one before to adjust to the new organism. What that eventually did was that it allowed for similarities between species, making our brains very similar to those of other mammals, or even vertebrates. If you look at these drawings, these are drawings of 19th century German zoologist Ernst Haeckel. He drew similarities between vertebrate embryos. So on the left here is a fish. So these are earlier stages of development, and these are later stages. And all the way down here are human beings. You'll notice that early on in fetal development, the embryos look quite similar. That attests to the strong evidence that we shared a common ancestor. Although Haeckel's theory received strong criticism during his day, we believe that this conserved evolution allows us to be able to use animal models to better understand the human brain. In fact, the brain is quite similar between species. On the left is the hippocampus of a kitten, and on the right is the hippocampus of a human being. If I told you, if I didn't have these labels, you probably wouldn't be able to tell the difference. But even to an untrained eye, hopefully you can observe the similarities between these two very different mammalian species. Interestingly, the development of the cortex, which is not depicted here, is very similar between humans and birds. So the fact that these similarities exist not only supports the theory of a common ancestor that Haeckel was advocating for, but also supports the idea of a similar genetic control of brain development across species. What we have here are three species, three rodent mammals, and a researcher identified the areas of the brain in these three species that handle touch, hearing, and vision. What you'll notice is that these areas of the brain are roughly in the same location across these three species, but what you'll notice is that the size of these structures are very different between the three. That's because they rely on these senses differently from each other. For example, the mouse, which is a nocturnal animal, relies very little on the visual system because it's dark, rather relies on its somatosensory system or touch. The same is true for the vole, and so the somatosensory cortex is much larger in this structure than it is in the mouse. The opossum relies on its sense of vision to gather information from the environment, and so the area of the brain devoted to vision is much larger than that of the vole or the mouse. But the idea holds true is that there's conservation of these structures across these species and supports the use of animal models for studying human brain development. Interestingly, not only are neural structures conserved across mammals, but behaviors also seem to be conserved across mammalian evolution. Playful behavior is one of the most prominent behaviors observed in young mammals across species, from rats to chipmunks to monkeys to humans. You might have even observed some rabbits in your yard playing around, chasing each other. Rat pups engage in distinctive patterns of play, including object play, movement play, play fighting, or even rough and tumble play as depicted here on the left. While they play, they emit chirps that are emitted during socially rewarding situations such as sexual encounters. The exact function of play behavior remains unknown presently, but researchers have suggested that it might help animals learn about their physical and social environments. Play consumes as much as 20% of an animal's activities and up to 10% of its daily energy expenditure. It can be dangerous as it makes them more easily noticeable. For example, juvenile fur seals suffer their highest mortality rates when playing because playing in the water often makes them more vulnerable to predators. And yet the drive to play appears to be incredibly strong. Play behavior typically diminishes in rats as they reach puberty, and although they continue to play in adulthood, it's at much lower levels. Let's talk about the brain areas that might be involved in play behavior. The neocortex, which is the most recently evolved structure, doesn't seem to be involved because removal of the neocortex has no impact on play behavior, suggesting that play behavior might be controlled by subcortical structures. So the mesolimbic dopamine pathway, which we've talked about before in a previous lecture, which is the reward pathway, seems to be important for many reward aspects of play. Certain areas of the thalamus seem to regulate somatosensory or touch input. Periaqueductal gray seems to coordinate play responses. And the striatum, which is not listed here, seems to influence intentional movement patterns. Let's talk about the chemicals. It should come as no surprise to you that dopamine is involved, as it is a key chemical in reward. Endogenous opiates, which are released from the periaqueductal gray, seem to also be involved. Serotonin, interestingly, decreases with more pins during play fighting from a playmate, and growth factors, which promote brain development, seem to increase with play behavior. Interestingly, psychostimulants such as amphetamine and methylphenidate, which is Ritalin, reduce play behavior. This makes you wonder about the impact of ADHD medications. Interestingly, given the effect of amphetamine on play behavior, a researcher from Washington State University, Jaak Panksepp, decided to look at the relationship between opportunities for children to engage in play and the prevalence of ADHD, which shown here has increased dramatically in both males and females since 1998. He also observed that rats deprived of play exhibit an increased desire to play. So Panksepp thought perhaps play-deprived children may also experience such play urges, a response that adults may interpret as a form of impulse control disorder, or ADHD. Now, ADHD is definitely a complex disorder, but Panksepp, I can't say his name right, suggests that some cases might be influenced by the child's social and physical environment, with decreased opportunities to engage in play leading to increased susceptibility in ADHD symptoms. Let's take a look at how play behavior has changed. So when researchers used a representative sample of mothers and asked them to compare duration of outdoor play of them as they were as children versus their children, mothers, 70% of them spent most of their day engaging in play, compared to only 30% of their children engaging in play. So given this drive for play behavior, it's clearly important for the developing brain, and we need to consider how much time we allot for children to play outdoors compared to what they have in the school day versus in the summertime. Are children really spending time outdoors playing the way they should to promote brain development, or rather are they spending it indoors, engaging in more passive behaviors such as watching TV or playing video games? We're going to shift gears a little bit and talk about something that we've discussed before, which is epigenetics, but delve into it in a bit more detail. So I want to highlight the fact that the choices we make in life, our environment, can influence what is inherited by our children. So we know that the environment plays a huge role in brain development, but perhaps you hadn't considered that your grandfather's environment may actually influence your brain development through epigenetics. Again, we've discussed epigenetics before, but we'll revisit it. But first I wanted to tell you a little bit about Lars Bringen. I can't say any of these names. Bygren, Lars Bygren, and the northernmost country in Sweden called Norrbotten. So Norrbotten is in in Scandinavia. It's freezing cold there, and Norrbotten's different because it's found in the northernmost portion of Sweden, and it's home to only 6 people per square mile. So the number of people in this area of Sweden is even less. During the 19th century, this territory was so remote that in years of poor harvests, the inhabitants of this region ended up risking starving to death. The stress associated with these crop failures was exacerbated by their unpredictability. Historical records indicated that starvation occurred during specific years. That was 1800, 1812, 1821, 1836, and 1856. But there were also years when crops were bountiful. And those years of bountiful crops were 1801, 1822, 1828, 44, and 1863. The crops were so plentiful during those years that residents were able to gorge themselves. All right, so fast forward to the 1980s when Lars Bygren wondered about the long-term effects of poor harvests on the survivors of Norrbotten's starvation years, and if these famines had any effect of subsequent generations. So contrary to Darwin's idea that changes in DNA take millennia and occurs over long terms of time, Bygren's work suggests that actually we aren't born with a clean slate of DNA. Let's take a look at what he found. Boys who switched from normal to gluttonous diet, this is during the years of famine to the years of bountiful crops, ended up having sons and grandsons that lived shorter lives. Paternal grandfathers who experienced 2 extremes of feasts and famines lived 32 years less than their comparative cohort, the ones that didn't experience the 2 extremes. What seems to be the end result is that transgenerational effects appear to result in reduced longevity and increased the risk for diabetes and cardiovascular disease. What this means is that even if you have a very, very healthy lifestyle, the lifestyle of your grandparents or your parents can influence the likelihood of your survival as well as your risk for developing diabetes. So again, this idea that genes are a clean slate when we're born is not the case. Lifestyle choices made by our parents and grandparents can ultimately affect our physiology and behavior. Let's take a look at a study that was done in rats to demonstrate similar findings. So these important clues about transgenerational findings have also come from rodent research. So a researcher in Canada, Michael Meany, and his colleagues conducted a study on maternal behavior on offspring. So you might not know this, but rats typically lick or groom their pups as part of their maternal care. And Meany noticed that some moms lick their pups more than others. And so what he did was he kind of grouped moms into high licking or low licking categories. And then he tested the offspring from the high licking mothers and low licking mothers. He found several differences. First, he noticed that offspring that came from moms that licked a lot had reduced levels of stress hormones and more receptors for these stress hormones in their hippocampus, What this means is that the hippocampus is more sensitive to the stress hormone, and the hippocampus usually shuts off the stress response. So when we get stressed out, cortisol gets released from our adrenal glands, and the hippocampus works to shut off the system so we don't have too much stress hormone in our body. The more sensitive, the more receptors the hippocampus has, indicates that the hippocampus has greater sensitivity and therefore shuts off the system quicker. Interestingly, adult offspring of the high-licking mothers also exhibited diminished startle response, so they didn't get as startled as easily, and they were also less nervous, demonstrated by greater exploration in a new or novel environment. So we know that it's not the genes, but the environment that the moms are creating for the offspring to generate these kinds of differences in physiology. So Michael Meaney, to kind of argue that, oh, the high-licking mothers aren't just passing on the, good genes to the offspring, they created what's called a cross-foster experiment. So they took the pups from low-licking mothers and gave them to the high-licking mothers. So the mothers would lick their pups again. So now these moms do not share any of their genes with their offspring, but all the moms are doing is caring for the pups the way they would their own. Interestingly, rats, as Michael Meaney predicted would happen, behaved as their fostered mothers rather than their biological mothers. So what they found was that the rats raised by high-licking mothers, whose biological mothers were low-licking, were less fearful and less afraid compared to those who were raised by low-licking mothers. So this suggests that the care that the mothers are giving are changing the brain of the offspring, resulting in different behavior in adulthood. Another experimenter used this high-licking and low-licking paradigm, and he tested the anxiety response of the offspring using this behavioral test called the elevated plus maze. So there are closed arms shown here and open arms. Most animals, if they're really scared, will spend more time in the closed arms and not as much time in the open arms, because the open arms kind of leaves them to be more vulnerable. So if they spend more time in the open arms, though, it indicates that they're less anxious or less stressed than if they spend time in the closed arms. So let's take a look at how the high-licking and low-licking offspring differ from each other. So the low-licking are in purple, and green are the high-licking offspring. And so this is the central area, which is kind of like the neutral area and typically where they're placed. The open arm is the one that indicates they're less fearful. The closed arm indicates that they are more fearful. And what you have on the y-axis is the amount of time that they've spent in each of these areas. So the high-licking group spend more time in the open arm compared to the low-licking group, and then the low-licking group spent more time in the closed arm, suggesting that they're more afraid than the high-licking group. So this suggests that the offspring, the way that the offsprings are raised, impacts their brain function and behavior later on. So we kind of talked about this before, but epigenetics is the tags modified or added to DNA to change how it's expressed. Some of these chemical markers are shown here. They include methyl group, they include acetyl groups, as well as phosphate groups. You don't need to know these specifics, but what you do need to understand is that tags on the DNA or on the proteins that the DNA is wrapped around changes whether the gene will be expressed or not. These tags are added on based on environmental signals. They are not something that is automatic. So environmental signals determines whether these tags are added on to DNA or the histone proteins that act as spools on which DNA is wrapped. But these tags can be passed on from generation to generation. So these epigenetic mechanisms enable the permanent code, which is in the DNA, to be transiently tweaked to enhance survival in a changing environment rather than waiting for the global changes that occur with evolution in the genetic code itself. We're going to shift gears and talk about neural development for the rest of this lecture. So as neurons are born, they are positioned within functioning neural networks, allowing animals and humans to be able to breathe, eat, walk, as well as navigate life's many, many challenges. One of the first steps of brain development or neural development is the formation of the components, the basic component of the central nervous system. The way the central nervous system forms is through the process of neurulation. So initially, the embryo is just a ball of cells, but then it gets more and more specialized. So what you're viewing here in this first group of pictures is the dorsal view of the embryo. So this is the top of the head, and this is what will become the base of the spine. And the neural tube, which is the basis for the central nervous system, is essentially just a tube that is formed through the folding of the outer edges of this initial stage of development, and it zips up to create the tube. So what you're looking at here on the bottom images is a coronal view. So if we were to slice through the developing embryo, this is what you would see. The neural tube forms in the outermost layers of cells, and you don't need to worry about these terms so much, but notice that the sides end up rising up to come together to form the neural tube. This green diagram here, this green structure here, eventually forms the central nervous system, which is the brain and the spinal cord. And what essentially happens during development is as more and more cells form, you get more specialization within the tube. So here is the neural tube. Oops, here's the neural tube, right? This is rostral, this is caudal. Again, don't worry about the terms here. Oops, don't worry about the terms shown here, but you should recognize forebrain, midbrain, and hindbrain from the previous lecture that we discussed. But essentially, when the neural tube forms, you end up with growing larger structures that eventually become the forebrain, midbrain, and hindbrain, and this last component here is the spinal cord. The internal components, shown here, become the chambers or the ventricles of the brain. So here is greater differentiation of the forebrain. You should recognize that these are the hemispheres that are forming around the forebrain. And you have the diencephalon, which will eventually become the thalamus and hypothalamus. But what you should note is that the optic vesicles, right, they eventually are part of the brain, they eventually reach out to join with the eyes. You might think that the eyes are not part of the brain, but in fact, they originate from the brain. So your retina originated in your brain, but joined up with the eye during brain development. So here's kind of the image of brain development from your textbook. You'll notice that really all it is a tube that gets more and more specialized over the course of development. By 28 weeks, you can recognize that this looks like the human brain, although it's very, very smooth. But by 36 weeks of development, the fetal brain looks very, very much like what the adult brain looks like, with all the convolutions found within the adult brain. So let's take a look at what happens between the initial neural tube stage of development all the way to the end of fetal development. So here are the 6 stages of development: neurogenesis, migration, differentiation and aggregation, synaptogenesis and circuit formation, apoptosis, synapse rearrangement. And we'll talk about each one of these steps in greater detail in the coming slides. So the earliest stage of development, right, so neurogenesis, if you're not familiar with the term, is the formation of neural tissue, more specifically neurons. Gliogenesis is the formation of glial cells. So in the earliest stages of development, even before the neural tube has formed, cells that are dividing are what are known as pluripotent. These cells are able to form any different type of cell in the body. So they're considered to be stem cells. You might have heard of the term stem cells in the media. But they are pluripotent, which means they have the potential to become any type of cell in the body, a heart, a liver, a muscle, and even brain. But following neurulation, these cells in the neural tube are more specialized and give rise to specific cell types. They can become any cell in the brain, so they're considered neural stem cells, but they are not pluripotent. So these are progenitor cells that give rise to neural stem cells. So neurogenesis, as I mentioned to you, is the formation of new neural cells or neurons, and gliogenesis is a formation of glial cells. Neurogenesis occurs in specific areas of the neural tube. It doesn't happen everywhere, but it happens in specific areas of the brain known as proliferative areas, and they happen to be in the granular zone of the hippocampus and the subventricular zone. So I'll point that out to you here. So the subventricular zone I'm drawing out here resides right up against the ventricle. So this is the ventricle of the neural tube, and all neural tissue are born in the subventricular zone. But these neural cells then migrate from the subventricular zone to their final destination using radioglial cells. So neuroblasts are the newly born cells shown here. They're baby neurons. They travel to their final destination on radioglial cells. So radioglial cells, we've talked about them before, only are found during development. They have their cell bodies in the subventricular zone shown here, and then they extend their arm all the way to the edge of the growing brain. Neuroblasts then migrate on these radioglial extensions, kind of like the yellow brick road in The Wizard of Oz, and then hop off these extensions when they get to their home or final destination. After development, radioglial cells turn into astrocytes, one of the many support cells we've talked about in the previous lecture. Once neuroblasts get to their home, neurons aggregate to form collections of brain areas. So on the right here is a stained human brain section. You'll see that there are collections of blues and purples. It's not evenly distributed within the sections. So for example, right here, you can see that there's a collection of brain cells, right? Same here, right? These are neurons that aggregated during development to form these individual structures. These aggregated neurons then work together to form circuits within the region to perform a particular function. So remember we talked about the substantia nigra a while back when we were talking about neuroanatomy? These neurons aggregated during development to form the substantia nigra and then differentiated into dopamine-producing neurons, or they turn into glial cells that reside within the substantia nigra. Differentiation is when a neuron takes on a particular identity. Here you have neurons that are found in the cortex. They are known as pyramidal cells because of the triangular shaped cell body. These neurons get their specific shape based on cues from the environment that pull or push the dendrites upward and pull or push the axons outward. Incredibly, these axons of these neurons from the cortex will traverse long distances to eventually end up in the spinal cord. Makes you wonder, how does this happen, right? It's the process of axon guidance. So a neuroblast takes on the appropriate position in the nervous system after it differentiates and extends its axon and dendrites, it sends out what's known as a growth cone. Growth cone is like finger-like extensions. It's a growing tip of what will eventually become the axon. It's specialized to identify the appropriate path for an axon to elongate. It's got these projections called lamellipodia or filopodia. Again, you don't have to worry about these terms so much, but recognize that these projections probe the environment. They kind of feel out the environment to decide which way they're going to want to go. And here is an actual diagram, an actual picture of a growing axon. So this is the axon, and this is the growth cone. It's actually extending out in two different directions to sense out the environment to determine which way it's going to want to go. Based on the cues it receives, the axon is guided to its particular destination. So axons traverse massive distances in the adult brain. So as I mentioned, areas in the motor cortex, for example, reside in the cortex, and they end up reaching all the way to the base of your back to synapse with motor neurons that reside in your spinal cord. So the common pathway is formed by pioneer axons that we're not going to worry about, that find the way, and these axons end up following molecular signals in the environment called guidance cues. The growth cone contains receptors for these guidance cues to detect them in the environment, but these molecules affect the rate and growth of the growth cone. So these filopodia, these finger-like projections, grow out and sample the environment and decide which way they're going to want to go, and then the cues that they receive determine which way they go. There are two types of guidance molecules. There are chemoattractants, which attract the growth cones towards them, but then there's also the chemorepulsants, which force the growth cone away from them. So let's take a look at an example of what I mean by these chemoattractants and chemorepulsants. You don't need to know the specifics of these examples, but it's just to kind of demonstrate to you how the growth cone uses these attractants and repulsants to grow towards or away from them. Here is the spinal cord of an animal model, a fruit fly. You don't need to worry about that, a fruit fly. There are two molecules that serve as chemoattractants and repulsants. Netrin is a chemoattractant. SLIT is a chemorepulsant. Both of them are expressed by the midline, the middle of this growing spinal cord. So a growth cone expresses netrin receptors, so it grows towards the middle of the spinal cord, or the growing spinal cord, but then it crosses over the midline. Once it does that, it expresses the receptor for SLIT. And remember, I told you SLIT is a chemorepulsant. Now, what's going to happen is this growth cone is going to grow away from the midline rather than crossing over and over again. So in this way, the molecules in the environment pull the growth cone towards it or push them away from the midline. Through the process of axon guidance and differentiation, neurons begin to form synapses and circuits. They form connections with brain areas and start forming synapses. They begin releasing neurotransmitters to see if the synapse is active or not. This is the basis for all aspects of a functioning nervous system. But not all synapses are accurate early in development. In fact, when an axon reaches a target brain area, it forms connections that are more diffuse, shown here on the left. So when it reaches its target, it kind of synapses randomly and diffusely, but following activation of synapses through the matching of specific neurotransmitters and the receptor, the synapses get rearranged to be more specific, as shown in the diagram here on the right. This process involves the release of neurotrophic factors. Rita Levi-Montalcini was a brilliant neuroscientist who worked in Italy even under the control of the Nazis. So when she couldn't do her research at her university, she kind of took her research to her bedroom. She was the one who first discovered these molecules called neurotrophic factors. Neurotrophic factors are essentially food for growing neurons. They promote brain development. She discovered the nerve growth factor, and she found that when the nerve growth factor was given to new neurons, they sprouted all kinds of synapses and dendrites and axons. So she recognized this molecule promoted brain development. Later on, they identified another neurotrophic factor, brain-derived neurotrophic factor, that we now know is critical even in the adult brain to promote things like learning and formation of new synapses for memory. So I just wanted to highlight Rita Levi-Montalcini. She's a big name in neuroscience, and she was the first one to discover the nerve growth factor as well as the idea of neurotrophic factors. So let's apply this to what we talked about before, which was synapse rearrangement. So neurons that form synapses accurately with other neurons end up taking up neurotrophic factors. These neurotrophic factors keep neurons alive. So early on in birth, this is what the brain kind of looks like, sparse connections. At around 7 years of age, there's a lot more connections that occur as a result of those growth factors. But as connections get more and more specific, there's something called pruning, where the synapses that are not specific die off and the ones that are more specific are retained. The ones that are retained take up neurotrophic factors and are kept around. This leads to the process of apoptosis. So neurons that don't form synapses with other brain regions are not kept around. They don't get neurotrophic factors, and so they don't stick around. Instead, they end up being killed off through a process known as apoptosis or programmed cell death. And so it's a given that the developing brain, is going to make more brain cells than it needs, with the understanding that the ones that are not used are going to end up dying off. So it's thought that about 50% of the neurons that are made during development end up dying off during development and aren't used, and only 50% of them are actually retained. The very last part of development that your textbook doesn't highlight, but I think is kind of important, is the process of myelination. Myelination makes for a more efficient brain. And so myelination is a process of wrapping the axon of a neuron to allow for quicker conduction of the action potential. As you know, Schwann cells wrap or myelinate axons in the peripheral nervous system, whereas oligodendrocytes myelinate axons in the central nervous system. The process of myelination is very, very long and can last for up to 30 years. What you're looking at here is myelination, and from gestation, which is fetal development, all the way through 30 years. Myelination is complete in the first few years in sensory systems, but take a look at areas that handle much more complicated functions. Myelination in these structures does not occur until about 30, or it doesn't end until about 30 years of life. So your brain is currently still developing if you're under 30 years of age. This brings us nicely to the next phase of development, which we'll be discussing in the second lecture of this week, and that's the adolescent brain. So for now, let's summarize this lecture. Brain structures and behavior, play behavior and more specifically, are conserved across species, allowing us to use animal models to study the human brain and behavior. Environmental effects on our genomes can influence the physiology and behavior of our offspring through the process of epigenetics. And brain development has multiple precise steps that occur, the first of which is neurulation, or the formation of the neural tube, from which all neurons are generated through neurogenesis. Following neurogenesis, neurons migrate to their final destination using radial glial cells and then ultimately differentiate in that location into a specific cell type. Through axon guidance, they find their targets, and then through synapse rearrangement, they get more specific synapses with their target neurons. Neurons that don't form connections with other brain areas die off through apoptosis, resulting from a lack of neurotrophic factors such as nerve growth factor. Myelination, which is the last portion of brain development, lasts decades into your life. So that's it for this lecture. Don't forget to submit your reflection.